So, hello and welcome to the session. Here we're going to talk about electron microscopy. So, this is in continuation to the microscopic techniques which we have studied earlier. The previously mentioned techniques of microscopy, they were based upon how light interacts with a specimen and produces an image. So, the image of the object was produced by using light. So all these different techniques like bright field microscopy, dark field microscopy, phase contrast, confocal, fluorescence, all of these different techniques which we have studied, they were based upon the use of light for producing the image of the specimen. However, one important factor in governing the resolution and in turn determining the magnification which could be produced is the wavelength of the wave which is being used to produce the image. In case of light, the wavelength is relatively shorter as compared to the wavelength of the beam of electrons. Hence, by using a beam of electrons, we can achieve better resolution and hence higher magnification is possible. Such magnifications, they are not possible when we use light to produce the image of the specimen. So in case of electron microscopy, it uses a beam of electrons rather than a source of light to produce the image. The beam of electrons has a shorter wavelength as compared to the wavelengths of light, and hence it can produce higher resolution. So therefore, we can look at fine details of the specimen which otherwise are not visible through using other techniques of microscopy. So the significance of using electron microscopy is that the wavelength is shorter and hence the resolution which could be achieved is greater. In case of other microscopes which are based upon light, the lenses are made up of glass and they are optical lenses. So they converge the beam of light and help in producing magnification. In case of electron microscopy, the lenses are not made up of glass. Electrons are charged particles and hence they are affected by an electric field. So the lenses which are used in electron microscopy, they actually are electromagnets. By varying the intensity of electromagnetic field, we can we can focus the beam of electrons onto the specimen. Analogous to the manner that we focus light onto the specimen by using glass lenses. So in case of electron microscopy, the lenses are actually electromagnets. So under the influence of this electric field, the electrons are going to be focused in form of a beam and hence they are allowed to interact with the specimen. So the lenses in case of electron microscopy are actually electromagnets. The sections of the specimen which are produced for electron microscopy are very thin and hence, of course, the colors are not visible. So in order to produce a contrast, stains are used. In case of microscopic techniques where we use light to produce the image, we use different types of chromogenic or fluorescent substances as stains to produce a contrast. For example, in case of bright field microscopy, we use several different type of stains which are colored dyes. They bind to different parts of the specimen or the background and hence they produce color. So therefore, the specimen is visible as compared to the background. For example, you would have used different types of staining agents to look at the cells. So in case of electron microscopy, the staining agents are electron dense substances. Most of them, they are salts of heavy metals. Now, heavy metals, they are electron dense substances. So what is done is that the specimen could be coated with a layer of the salts of these heavy metals. So substances like silver, iron, gold, uranium, they could be used to produce a covering, a layer onto the specimen. Now when the beam of electron is incident onto the specimen, which is not stained, the beam of electrons cannot interact with the specimen properly. However, when the specimen is stained or coated with these electron dense substances, then the part of the specimen which contains the electron dense substance is going to interact and deflect the beam of electrons. When the beam of electrons is deflected, this is recorded by a detector. And based upon the nature of scattering, the angles of scattering, a particular image is produced. All this data is recorded onto a computer, and then the data is processed, which in turn is used to produce the image of that specimen. Since no light is involved in the process, so the image of the specimen which we get, that is a black and white image. As compared to other microscopic techniques which we have studied previously, they can produce a colored image of the specimen depending upon the color of the fluorescent molecule or the dye which is used for staining. As opposed to those methods, since there is no light involved in the process here, so electron microscopic technique, it produces images which are black and white. However, by using image analysis softwares, image modifying softwares, we can impart colors to different regions of the specimen. So those colors are a result of image processing. Those are not the actual colors of the image which is produced through electron microscopy. The two important methods of electron microscopy are transmission and scanning electron microscopic techniques. So now we are going to look. The first one is transmission electron microscopy. In this method, there is a source of electron. As you can see in the diagram, this is an electron gun. The electron gun is a source of electron. Different type of substances, they could be present in the electron gun. For example, it can contain tungsten or other different types of metals which produce, which when stimulated, they produce electrons. So they produce electrons which are released in form of a beam. Then here you can see this is a condenser lens. As in case of a light microscope, the function of the condenser lens is to converge the light rays in form of a narrow beam so that it is going to be focused onto the specimen. In case of an electron microscope, the condenser lens does the same function, that it collects the beam of electrons and converges it onto the specimen. However, this lens is not a glass lens as we have seen in case of light microscopes. This lens is an electromagnet. So when the electrons, they move through this chamber, through the electron microscope, so they influence this electric field produced by the condenser lens, which is an electromagnet. Under the influence of that electric field, the electrons are going to converge in form of a beam, and then they are incident onto the specimen. So the electrons, they pass through the specimen, and then upon interaction with the specimen, they are deflected. These electrons are then collected by another electromagnet. This electromagnet is the objective lens. So it collects the information in form of the electrons which are deflected from the specimen. So some electrons would be able to pass through the specimen, others they will be deflected by the specimen. Here, in order to improve the contrast, we add the stains, and the stains are electron dense substances which form a coating onto the specimen so that the specimen becomes stained with the electron dense material and the background is not stained. So this is called as positive staining, where the specimen is stained but the background is unstained. So now the regions of the specimen, say the background, which are not stained, the beam of electrons are going to pass through them in a straight manner. However, the part of the specimen where the stain is present, the electrons are going to be deflected. So these electrons are in turn collected by this electromagnetic objective lens, and then these beam of electrons are used to produce an image on a detector. The detector could be a photographic film or a fluorescent screen. This is connected to a computer, and by using a computer software, the pattern of the electrons is detected and stored. And based upon the pattern and the way in which the electrons are going to be collected by the fluorescent screen, that is then used to produce the image of that specimen. Here you see an image of a cell which is obtained by using transmission electron microscope. So you can, since the beam of electrons is able to pass through the specimen, so you can look at the inner details. Also, a very thin slice is needed for producing a transmission electron microscopic image. So therefore, most of the information which we get through transmission electron microscopy is providing information in a two-dimensional plane. So the resolution is of the order of 10 picometers, and it can produce magnifications up to 100,000 times. When the specimen is stained, it is coated with an electron dense substance that is called as positive staining. Staining could be done in the opposite manner as well. That is, the background could be stained and the specimen remains unstained. This is called as negative staining. Since it is a very thin section which is used to produce the image, so the information we are getting is in form of a two-dimensional image. The specimen, it has to be fixed, it has to be dehydrated and is processed under vacuum. It is important to have vacuum so that the beam of electrons is able to interact with the specimen properly. So because of these reasons, that the specimen is fixed and therefore it is killed, it is no longer alive, it is dehydrated and is operating under vacuum, so that can result in shrinkage or other modifications of the specimen. And sometimes this can produce artifacts as well. However, the significance is that the resolution and magnification obtained by transmission electron microscopy is much higher as compared to any other microscopic technique we have studied. So this is how the image would look like. The image is actually a black and white image which is later colored into by using these different colors by using image processing softwares. So the transmission electron microscopy provides information mostly about the two-dimensional view of the specimen, and also the processing of the specimen can lead to producing artifacts as well. So these problems they could be solved by using another type of electron microscopic technique, which is called as scanning electron microscopy. In scanning electron microscopy, the electrons are not going to pass through the specimen. So what happens here is that in a similar manner, there is a source of electrons which produces electrons. The electrons are focused by using electromagnetic lenses, and then the beam of electrons is incident onto the specimen. These electrons, when they are of relatively low energy, so when they fall onto the specimen, they collide with the atoms of the specimen and they knock out electrons as well. So the electrons which are coming through the source, they're called as the primary electrons. They interact with the specimen and they release electrons from the specimen. So based upon how an incoming electron hits the specimen at what angle and what type of electrons are being emitted from the specimen, so that information is collected by this detector here. So the primary electrons which are coming from the source, they strike the specimen and then they knock out electrons from the specimen, which are called as the secondary electrons. Then these secondary electrons are collected by the detector, and in turn this information is used to produce the image of the specimen. As opposed to transmission, scanning electron microscope can produce a resolution of the order of tens of nanometer, and the magnification that could be achieved is of the order of 10,000 times. However, this generally uses a relatively thicker section of the specimen and hence is able to produce a three-dimensional view of the specimen. So this is an image of the cell which is obtained by using scanning electron microscope. Here in this picture from the book, you can compare the images which are produced through transmission electron microscope and scanning electron microscope. So you can see here that the section is thin. It produces a two-dimensional image of the specimen, and therefore we are looking at a certain plane and it provides information about the internal structures as well. The image obtained through scanning electron microscope generally provides information uh, for a thicker section, hence produces a three-dimensional image and provides information about the surface of the specimen. So all these different images which are visible here, so they are representing different orientations of the same specimen. So multiple images of the specimen are collected, and then by using powerful imaging softwares, the specimen molecules which are in the same plane, they are collected. So these boxes are representing the molecules of the specimen. All of these are the same type of the molecules. So the boxes represent the molecules which are in the same plane, in the same orientation. So this information is collected and stored by using the software. In this manner, we get a unified image for many different orientations of the molecules of the specimen. And then all of those images which correspond to one particular orientation of the specimen, they are combined together to produce a unified image. So here you see the same specimen is in different orientation. So the images were collected from that raw data, and then all those selected boxes, they were used to produce the image of the specimen in one orientation. This is corresponding to the set of images which were obtained for another orientation of the specimen. The next one is a third orientation, the fourth orientation, and so on. So multiple images of the specimen which are in one plane, they are unified by using an imaging software, and then it produces a map of the electron density and which is collected, and the images are unified to produce one particular orientation of the specimen. In this manner, we get a series of unified images, each of them corresponding to a particular orientation of the specimen. Then all of these different images representing various orientations of the specimen, by using the computer softwares, all of these orientations are compiled with one another. In turn, it produces a three-dimensional image. Here in this picture, you see the raw data, the image. This is corresponding to the flagellar basal body. Flagella are structures present in bacteria which are used for movement. So this is the structure of the basal body. The basal body is the structure of the flagellum which is embedded in the membrane. So multiple images of the flagella which are lying in one plane are collected, and then they are all unified to produce um, these averaged images. And then these images are used to reconstruct the three-dimensional structure of the flagellum. So that is diagrammatically represented here. So the flagellum is made up of multiple proteins. So this is how the electron density map is going to look like, and then the proteins which the individual proteins which form the flagellum, their structure is shown in this representation, is fitted into this electron density map. In this manner, this could be used to reconstruct the three-dimensional image of this macromolecular complex. So one important significance of electron microscopy is that the resolution is much higher as compared to other microscopic techniques. The magnification which we produce is also of the higher order as compared to other light-based microscopic techniques. And another important significance is that a much detailed three-dimensional structure of the proteins or other molecules or other structures could be reconstructed by using electron microscopic techniques. So these are some of the important aspects pertaining to electron microscopy.